Hello.
Yes, Tripathi, sir.
Dr. Vinod, I am audible?
Yes, you are audible, sir.
Okay, thank you.
Just a minute, sir. We'll go.
Yeah, please.
Good afternoon, sir.
Good afternoon. Torno, how are you?
I'm good, sir. How are you, sir?
I have just run a bit of the interview. Chalo.
Nice
Just
to
15,
see you,
20
sir.
minutes. Yeah. You have to permit me to share the slides.
Yes, we'll be doing that, sir. We'll be doing that. Live on YouTube, and you can share your slide. Meantime, Inyan will introduce you to the gathering. Thank you.
Good afternoon, everyone. Welcome back to the afternoon session of the International Conference on Bacteriophage Research. So it is my privilege and honor to introduce our first speaker of the afternoon session, Dr. Upendra Tripathi. He obtained his PhD at the Indian Veterinary Research Institute, Uttar Pradesh in 1990. And then he served as a scientist at the ICAR NRC on equines and Indian Veterinary Research Institute, CSWRI, Avika Nagar as head division of animal health. He is also served as the director, National Institute of Animal Health from 2012 to 2014 and ICAR NRC on equines from 2014 to 2020. He is currently serving as Deputy Director General, Animal Science at the International Indian Council of Agricultural Research Headquarters, New Delhi. He has worked extensively on pathology and diagnosis of infectious diseases and received several research grants from various funding agencies. He has published more than 200 research papers in national and international journals with 2000 citations, 3 books, 1 manual, and edited 3 conference proceedings. He had made pioneering contributions on molecular pathogenesis and diagnosis of John's disease. He is serving as a reviewer and editorial board member in many international journals and held many honorary positions. With these few words, I invite him to present his lecture on application of bacteriophage in veterinary sector and significance of bacteriophage repository in the current era of emerging antimicrobial resistance. Welcome, sir.
Now visible to you?
Yes, sir.
Okay, thank you. Thank you, doctor. I do not know your name. So thank you very much for
thank you, sir. Thank
introducing me to audience. And my colleague there in SBRT, Dr. Vinod, and another organizer of this international conference, second international conference of SBRT Society. And I must see Dr. Gopal and many other participants of this conference. And thank you very much for giving this opportunity. I thought that since we started work at NRC Pine National Center for Veterinary Type Cultures at Hisar, and we all together, Dr. Vinod and Dr. Gopal, Dr. Chibbar and others at BHU conference could be able to form this society. And I'm very glad to see that this society is going very well, organizing conferences, and perhaps it started a new era in the bacteriophage research in India, and also bringing people from several countries to come work and share their views and their work so that it can further become a reality, a real therapy for as an alternative therapy for microbials. So this is day 3 and the last day, and also the last session. And I could not listen everybody, but I could able to see that those slides that were presented by some of the speakers on day 2 and today also. I have found that there are very learned speakers from different other countries, and they were beautiful, very fundamental, and some of the application translation research that has been done on bacteriophage, and that was very good to see them. And they are not only so the use of bacteriophage we know very much, and there's no need to again emphasize that what are different uses and application of bacteriophages in the practicality. In fact, so only thing is that one of the lecture that was given by Dr. Martha from UK, perhaps. And she was saying that how closer we are to clinical deployment. That is the question today that everybody should ask. So with this background, I would like to start with, I'll just, first I'll start with AMR. Why do we need bacteriophage research and new unit therapy? And then how it is being applied in veterinary science and animal husbandry, and then switch over to at our bank of repository rather of bacteriophages at NCVTC. So friends, you can see that how the panoramic view of antimicrobial consumption in food animals. That is very important, and you can see that India is also one of them, and across the world, that the use of these microbials are being consumed for different purposes. And if you see that some data has been taken recently on some paper, you find that China is almost top user of antimicrobials, followed by USA, and then the Brazil, and then Peru, and the India, of course, the last. But also India, looking at the site, it also uses a lot of antimicrobials for different purposes. So, and here you can see the 2030 picture, that how it is going to be in 2030. So almost more than 100% increase for each country. So that will become the problem. And when Fleming discovered penicillin and antibiotic, then he raised this issue. And he apprehended that there could be possibility of antimicrobial resistance, and therefore those things are now coming in reality. You can see that how it is antimicrobial consumption, trend of consumption in livestock. That's up. So you can see. Look here, in cattle, in chicken and pig. The cattle, around 45 mg per piece is being used. This is statistical analysis. And chicken, it is 148 mg. So such amount of per kg of meat production. And then pigs, it is more than that. Even as a growth promoter, 172 mg being used per kg. So this is the view, that overview, that what is the problem and why such like MR issues are coming up in the country. And you can see on this slide that different countries, across they are using antimicrobials for different purposes across, China, and this India also using here, the South America and North America. So most of these countries that are using in a vast quantity, and that has really lead to AMR problem. And AMR is not only a problem, and it has been recognized by all the international bodies like FAO, WHO, and OIE, and everybody around the world. And we also in the ICR system working on the antimicrobial resistance and creating and generating a lot of quality data, so that policies, it can be a policymaker can take some call on it. So now this slide, just you can see that how extensive, what is that, how these antibiotics are being used, and how they are being transferred from human to animal and environmental and pharmaceutical industry itself. They're also causing a lot of problem, because when they dispose of their waste, they are coming in the environmental. So all these together, they are bringing AMR issue and creating problem for the public health issues. You can see here that how this resistance is spreads across the food chain and a human. This here the antibiotic, the animal gets, and from animals, these all get drug resistant bacteria that remains in the food chain. And once it comes in food chain, again it goes back to humans. And similarly, human can also get infection from the hospital and even to their colleague also. And finally they go to public. So this is a chain that how a very simplistic way to represent that how antibiotic resistance spreads among the animals and humans. So that is the problem. So, and also livestock, that I told that livestock, you are aware of that we have got a huge livestock, more than 536 million, and we have got 851 million poultry also. And for that we have been, and they are the top most milk producer in the world, and also fifth in egg production. And but those production, it is not without giving them a lot of growth promoters in the form of antibiotics. And so there, as a result, there are many bacterial resistant strains like Staph aureus, like E. coli, and salmonellosis, and multi-drug resistant bacteria are coming out of that. And that is where we have to really pinpoint and take care of it. So if you just look at what are the pattern of using antibiotics in animals, so they can be used for different purposes. Either you can treat of animals or treat of humans, or in control of a spread of infection. It is also used in the prevention of illness in healthy as a preventive and prophylaxis also being used. So this is all fine because we need to control the disease. But when it comes to growth promoters in the animal, livestock, and poultry industry, that is a lot. That all the growth, these promoters are being used in livestock, especially poultry and pig industry. So and they become the cause of antimicrobial resistance. So what are the options that we have with us? So that's why that now we are thinking of bacteriophages, and the people around the world are working on it. And when you come to the potential application of bacteriophages in veterinary sector, it is across the livestock, in the aquaculture and fisheries section, and also in the poultry. And all we have been using it. Some of the successful experiments have been done, and they really show the pathway that how we can go ahead and make a successful story for the fast therapy in some of these areas. So I have just reviewed some of these successful experiment that has been done in the livestock. So just I will very briefly, I will describe here that against Salmonella typhimurium in pig, again against, and also Escherichia coli and Salmonella, Staphylococcus aureus, and Escherichia clostridium pig. The fast cocktails have been used, and they have shown that how significantly bacterial count has been reduced, and that can be successful. Similarly, in cattle also some experiment have been done against the mastitis, like Staphylococcus dysgalaxia, and Streptococcus agalactiae, dysgalaxia, and uveris. These are the most important causes of bovine mastitis, one of the significant cause of reduced milk production. So against such, and this is the way that how antibiotics are secreted in the milk, and the milk is being taken by the humans. So this is how in the food chain antibiotics are being taken by the humans, children, etc., and leading to the transfer of resistance. So in this case also they have shown successful experiment in cattle. Similarly, in the weaning pigs, and again you can see some of the experiment that I have listed here. Against Staphylococcus aureus, and Salmonella typhimurium, and then also in the Bifidobacterium, Lactobacillus, and Clostridium, and Escherichia coli in pigs. They have developed a fast cocktail, and they have really shown the very good result of the improved gut health. And so because this can be a very good growth promoter also, why to use antibiotics? Why not to use these bacteriophages and see that the growth of livestock or poultry goes well. So these are some of the experiments. But the experiments are very new. Just last 10 to 12 years, this experiment have been done. So we can see that future is good. People are working on it, and they realize the importance of bacteriophage as an alternate therapy, at least in some of the cases, some situations where we can use it. Then also phage therapy in companion animals and dogs and equines, that it has also been used. And they have feel that One Health also is important from World Health perspective, and it could be alternative approach for phage therapy. You can see one of the experiment was Pseudomonas aeruginosa, where they given phage cocktail, and they found that electrolysis of Pseudomonas aeruginosa in ear without apparent toxicity. So this is no otitis. So this is the successful experiment. At least we can use some of the topical application. It can be used very well without much of the safety issues. So this is the first study, single study it has been done. Similarly, in the horses also, they have this keratitis and a pathogen against this Pseudomonas aeruginosa. In rabbit, experiment has been also done against Vibrio cholerae, and then again in dog against Escherichia coli and Pseudomonas, cat also. So these experiments shows that now people are showing interest, and also their results are worth taking to the clinical application also. So in poultry also, most of the poultry, you can see the meat production in the world is forecast by 2.6% to 136 million tons as per FAO. And majority of the infections in poultry are due to salmonellosis, E. coli, and Campylobacter. So that's also a great loss. And if such cases, if you are using antibiotics, that goes to food chain and ultimately results in the AMR. So some of the experiment that has been done in case of poultry in three weeks chicken, and they have resulted in the reduced morbidity and the severity. And you can see that they have also challenge with the Escherichia coli, and they have found there is a reduction in the mortality and shedding of E. coli. So in poultry also it has been so successfully, and similarly in quails, 33 days old cold quails, where they have again were a challenge with salmonella enteritis, and with almost similar MY infection with the bacteriophages, and they have found that there is a 100% efficacy in eliminating the salmonella infection from the tonsil. So that's also a successful experiment. Another experiment also, just to name them, the 47 days old broiler chickens against Campylobacter, and 1 day old chickens in salmonella enteritis, and the last broiler chicken against salmonella enteritis and typhimurium. So all these experiments, they really indicates towards its successful use, and it can be successfully used in the livestock and poultry industry as well. Now also in fisheries also, that people started using the aquaculture, especially against Vibrio Aeromonas and Flavorbacterium, Pseudomonas and Albertiella. So they're all that are very important organisms for the fish, and these there are successful experiment available for aquaculture also. You see some of the experiment against Flavorbacterium, Cyclophyllum, and then Vibrio parahemoticus, Shigella, and Cyclophyllum. So therefore, fish also, in addition to livestock, poultry, fish also it is being successfully used in experimental model, and this suggests that you can see that there are many papers that have come recently, even from different bacteria, furunculosis, vibriosis, or Aeromonas, and Advertiellosis, septicemia. So looking at all these application experimental studies, successful experimental studies, that shows that they can really of much help in that. One of the experiments that we did from the of isolating the a fast from my family engaged water. It was a thermotolerant and used against MDR and virulent Klebsiella pneumoniae in a murine model, and we found that it has successfully worked on that. And this is perhaps the first successful experiment against Klebsiella pneumonia in a murine model. So, and if you can see this is how there's extensive widespread neutrophilic infiltration and necrosis, and here is the consolidation and congestion. At the same time, in the treated group of animal, where it was treated with the same MOI, and we find that lungs have been cleared in 4 to 5 days. So this is a successful experiment that how we can use against Klebsiella pneumonia in murine model. Now the future prospect ahead, it is a biocontrol against aquaculture diseases. Although sustainability of fast therapy needs further investigation for the following, at least how we can use for the commercial application, that is important. And the formulation of bacteriophages for a layman's uses, though, and because every time technical problem is always there. So we have to look into that, how it can become a formulation that can be used by anybody. And development of protocols for various diseases with a consistent result. So there are some of the issues that we have to look into before we really deploy for the clinical application. Now the second part of looking at the veterinary application, there are pretty good examples and good experimental studies are available in livestock, poultry, and fisheries also. And hence, now our next step should be that how we can really go for the clinical application. And then I will discuss that how we should go for the official approval. There should be a legal guideline also to use them as a therapy, if not in all, at least in some of the situation and cases. So now my second part of my talk would be that we have got a bacteriophage repository at NRC Hisar, and where we are just working and collecting around perhaps 200 bacteriophages have been collected there and they have been preserved there. We are getting people are contributing also. And when we come to application part, then we really need an array of bacteriophages available. So that in any case situation, we can test them and supply to people who are going to work on it and who are going to use clinically. So as a phage biologist, prefer using lytic phages lacking any virulence and antibiotic resistance associated with toxin genes for pursuing the phage therapy. And the world status, if you see that there are a number of culture collections in other parts of the world, they have also started collecting bacteriophages, development repository bacteriophage also, and prominent among them are ATCC USA, and Biological Resource Center in Japan, and then DSMG in Germany, and the Netherlands collection of bacteria, Netherlands and China Center for Type Cultures Collection, and our center in India, that is National Center for Veterinary Type Cultures. So we need really to strengthen our culture collection so that it can be used by anybody from any part of the country. So this is our profile of NCVTC, and some of you might have visited. And also there was a workshop about 2 years back we organized with the help of ICMR, where Dr. Gopal was also came there and given a lecture also along with others. And many Urmi Bajpayee and others also were there. Dr. Chimbarwa also came there. So there we find that people are very interested, a lot of interest to work on bacteriophages. And perhaps we have started a very good thing in terms of establishing a society. And the society's work is now the major responsibility now, how they should see the phage research should go on, how they can collaborate, cooperate, and coordinate with each other. So that in addition to basic and fundamental research, we should also come to the clinical application of this. So this is some of the bacteriophage, the status that we have currently against all these bacteria. We have available bacteriophages, and anybody wants that they can also get it. We also accessing it, and this is approved by National Biodiversity Authority also. So our accession number is now very official, and it will also help you to get, if you make any patentable product out of bacteriophages, you need to deposit somewhere bacteriophage, because that's one of the mandatory requirement for applying for any patent. So that will also help us that such like research would further get a stimulus in form of having an official repository in India. Now, the major activity that we do at NRC is the bacteriophage isolation, purification, and preservation. and fast characterization by phenotypic and genotypic characterization, electron microscopic characterization. We also supply and demand, training and knowledge sharing. And the major challenges with us is how to maintain fast viability as a revival interval intervals. That is also a great job that we have to do. So Dr. Torna is there looking after bacteriophage repository. So she is struggling with, but even then she has been able to collect as many as close to 200 bacteriophages, and therefore it will further increase. And it could be a matter of interest for all of us that it should further grow. So this is in nutshell that what we really do. After isolation and characterization by transfer electron microscopy, pH, temperature, growth characteristic, host range, and then development of bacteriophage cocktail to cover range of bacterial strain, and dose selection, and then therapeutic trial. So we're also doing murine model. I've done now. We are doing some therapeutic trial in poultry as well. So therefore, I think this is what exactly at VTCC we are doing currently for isolation, characterization, and then preservation. These are some of the photographs of bacteriophages against E. coli, Bacillus, Shigella, Citrobacter, Pseudomonas, and Klebsiella. Now, what we see really is that we are committed to enrich NCBTC, and where we need your support from other part of the country, other laboratories who are working on that. And we assist fast characterization at genomic and proteomic level, that we have developed expertise in that. We're exploring fast therapy to combat bacterial infection in vivo and useful alternative to antibiotics to combat emerging antimicrobial resistance. And we always welcome to deposit bacteriophages for long-time preservation as a first master repository of phages. Now, this is one thing that I would like to tell here, that you may not able, those who are working on bacteriophages might be having number of phages in their collection, and you may lose it. I tell you may lose it in years to come. But this is ours, is a national repository, and our responsibility that we have, whatever we get it, we have to preserve it. And therefore there is a 100% guarantee that you will never lose it. You may lose it, but again you can get it from us. So therefore, my request to all of you, whosoever attending this conference and working on bacteriophage, they can also take it, and also they can enrich it, get their authentication, get the accession number, and it is a well acceptable, internationally acceptable repository now, perhaps. Now, some other thing that I would like to let you know, that now SBRT, our society has a great responsibility to work on the clinical deployment, and for that we need to have work together, draft guidelines, and get approval from the DCGI. We have to get approval. Dr. Gopal Nath very how a therapy, a drug can be licensed, or it can get approval of the government. So we have to work in that direction, and we may go on working on the basic and fundamental research. But alongside that, at least for some cases, in some situations where they do not involve much of the safety issues, for example, topical application, chronic wound, and maybe some of the, I think Dr. Gopalnath and others have done osteomyelitis and other cases. So at least there we can develop a guideline and a perfect standard operating procedure where we can use it. And we can start with some of the MDR, multi-drug resistant bacteria, and such cases problems comes every now and often. So such like cases you can do that, and then especially chronic diseases. Chronic disease, when you go for the systemic application of bacteriophage therapy, systemic means when you go for the parental dosing, then there are a lot of safety issues comes, a lot of immunological response comes. There we have to really study that how bacteriophages, when we are giving repeatedly are giving, then there is a lot of interaction with the host cell and bacteriophages, along with killing the bacteria. There are also many other immune responses that comes, how these bacteriophages are being processed by the host cell. So a lot of I did not see much of many of the papers on bacteriophages But my request would be that it is one of the area that the researchers should really explore it. That it is not going much of immunological problems in the long run. And then we have to go for the delivery system of. We can also use bacteriophages for different purposes, also as a diagnostic, as a vaccine delivery, and also as other many biocontrol, as surface disinfectant. There are so many applications that is possible. So my request and would be that let us work for its clinical deployment in some area of where the current therapy doesn't work. So this is all what I wanted to share with you. So thank you very much for giving this opportunity. I'm sorry that I could not give much time because I'm not at in Delhi. I'm somewhere else. So I have to find some half an hour time to come out of the meeting and then present this and interact with you. So with this, thank you very much. And if there are any questions, we'd like to answer them.
Thank you, sir. Thank you for the presentation in spite of your busy schedule. We have 1 minute for any discussion.
Dr. Tripathi, it was fantastic presentation, and we could see that the bacteriophages can be used in the veterinary sector, and somewhat we are doing also somewhere. But I think as clinical study we are doing, we should also focus to the veterinary. At least we can reduce the pathogenic bacterial load in the this animal husbandry sector, so that we are using less and less antibiotics. So it is really very great. And another thing is that your blessing is needed to hold another conference somewhere. So since you are sitting at the helm of the affair, I am sure that you will do something in this direction for the next conference. And we have to discuss this issue, but I think your blessings are needed.
Okay, I'm always with, because we have created this society, so I'm always with you.
Sure, sir. Sure.
This
Thank
was
you.
my interest itself. I'm very much interested since we started work with bacteriophage, and then you and Dr. Vinod and Dr. Chibber being the pioneer workers in the area. So we have to take this work forward. And I would request Dr. Gopal Nath to work on the clinical, at least, try to raise such like issues with those people who are at the policy maker level, right?
Yeah. Yes.
Because some of the topical applications we can very well use it, and those guidelines have to be drafted by clinicians, and it has to be accepted by DCGI.
Yes, sir.
If any country have got any guidelines, maybe like East European countries, they have got I think officially accepted therapy. So we need to take their help and how we can make guidelines, for our country, and because unless we have got some approval, right, official approval, then this research will not continue, right? Research will not continue. People will not like to work on that. So we have to, and there was a very good talk. I could not listen from Dr. Martha from UK. Clinical deployment is very necessary. So we have to work in that direction now. Thank you. All my question.
Thank you, sir, for a wonderful presentation. Since we are running out of time, and Mark Coleman is already inside for his presentation. Thank you.
Okay, yeah.
Indian, you can proceed with
Yes,
the next
sir.
presentation,
Yes,
please.
sir. Yes.
Okay. Thank you.
Thank you, sir.
I have to stop sharing, no?
Yes, sir.
Okay.
So we'll move to our next speaker, Dr. Mark Tolman. He is currently working as a senior lecturer at Cardiff University, UK. He received his PhD from Bristol University in medical molecular biology in 1998. And after working on host pathogen interaction with Haemophilus and Neisseria, he started working on antibiotic resistance in 2001. Since then, he has discovered and characterized several new and emerging antibiotic resistant mechanisms and mobile genetic elements. This work culminated in the discoveries of ISCR elements in 2006 and NDM1 in 2008. His interest in E. coli started with NDM1, the first metallo-beta-lactamase to give robust carbapenem resistant in E. coli. He moved to Cardiff University in 2007 and became a senior lecturer in 2013. He runs a small research group with the focus on using sewage to track antibiotic resistance. and sepsis rates, and is interested in the control of prevalent problematic bacterial strains by using bacteriophages to engineer the human microbiome. He has published about 100 papers, which together have received 15,000 citations. So today he will be presenting us the problem of virulent E. coli carriage in the UK and their control by bacteriophage. Over to you, Mark.
Thank you very much. I'll just try and share my screen here, which is not as easy as I thought it would be. Share screen. Okay. So let's escape that one and go back to here. Share screen, slides as virtual background. Okay. Screen. I'm struggling to share my screen here, unfortunately.
It is in the center of the screen. You can see there is a share screen, the green.
Yes, I'm pressing that, and then I get to somewhere else, which is not including my screen, unfortunately. Let me just switch briefly to another computer. If you just give me open system preferences. Okay. I'll just log off. I'll come back in a minute.
You can mail me your presentation. If you want, I'll share from my side.
He is not here, right?
Manju. No problem, I thought. ಬರೆಯಕ್ಕೆ ಶುರು ಮಾಡ್ಕೊಳ್ಳೋ ಅಪ್ಪ. ಜೆರಾಕ್ಸ್ ಕೊಡಕ್ಕೂ ನೀನು ಬೇರೆ 6 ಕಾಪಿ ಕೊಟ್ಟಿದ್ದೀನಲ್ಲಪ್ಪ. ಬರೆಯಕ್ಕೆ ಶುರು ಮಾಡ್ರಿ, ಅವರಿಗೂ ಬೇರೆಯವ್ರನ್ನ ಸೇರಿಸ್ಕೋ. Opinion? Sorry. Welcome back, Mark.
Yes,
Yeah,
really.
Please share your slides.
Really sorry about that. So, unfortunately, I've not got my backdrop on this one. But can you see my screen?
Not an issue. Please, you can take your time. Not an issue, Mark.
Can you see my screen? Okay.
Yeah, it is visible. Fine. It's good.
And if can you see that as well?
Yes, we can see the full screen, Mark.
Fantastic. lots of apologies for that. I'm terribly sorry. And thank you so much for inviting me, especially the members of the organising committee, Sanjay Chipper, Professor Vinod Kumar, Gopal Nath, Ramesh, Prasant, Urmi, and all the friends that I met last year or just the year before, actually, in 2019. It was a pleasure, and the conference has been really great. I've watched quite a lot of the presentations, and I hope to catch up on many others via YouTube. So I'm going to talk this morning in the UK, this afternoon, obviously in India, on the problem of gut carriage of virulent E. coli in the UK and their control or possible control by bacteriophage. And if we look on the World Health Organization site, they show us that back in 27, all 194 member states, the United Nations, needed to develop a national action plan against the condition sepsis, which is one of our world's most prolific killers. And basically on the World Health Organization site, they tell us that there's around about 50 million cases of sepsis worldwide. 20 million of these cases are in our children, less than 5 years old, shortening lifespans and causing misery. And then altogether, each year there's 11 million deaths from sepsis. And if we compare this to what's happened over 2020 with COVID, the World Health Organization have recorded 1.8 million deaths worldwide, and perhaps as many as 3 million. So every year, even without COVID, sepsis kills 4 times that number of people, and it's responsible for 20% of all deaths. So this really is a giant problem. And just advance the slide here. This is a giant problem, rather like Goliath, this 9-foot giant, a Philistine giant, back 3,000 years ago. And this young shepherd boy, David, who later became David, around about King David, around 3,000 years ago, he picked up 5 small smooth pebbles from a water brook, from a stream, a water source, and used them to take out this giant and stop the oppression that this giant was causing. So I honestly believe bacteriophages are capable of doing this. So in terms of sepsis, there are a couple of definitions. The lay definition is sepsis is a life-threatening condition that arises when the body's response to infection injures its own tissues and organs, or a more professional one, sepsis is characterized by a life-threatening organ dysfunction due to dysregulated host response to infection. And the title of this is Prevention is Better Than Cure. So this is using phages to prevent infection rather than actually using them as alternatives to antibiotics. And there are huge advantages in preventing infection before it occurs. These are the huge advantages. We reduce infection overall. We reduce sepsis rates and associated sepsis morbidities, such as the loss of limbs and loss of sight. We reduce the use of antibiotics, reduce naturally from that antimicrobial resistance rates, reduce death rates in general, especially for young children, and we reduce the huge society and healthcare costs. It's estimated in the United Kingdom, this costs the National Health Service between 1 and 2 billion each year, and the added costs society are estimated to be maybe as high as 15.5 billion UK pounds. So a huge amount of cost, financial as well as in people, and increase life expectancy and also increase life quality. So here are some figures of sepsis in the UK, and we definitely have a problem with sepsis. These are figures just England alone, so not including Wales and Scotland and Northern Ireland. And you can see here that back in 2010, there were about 92 1,000 cases of sepsis, and in 2017 to 18, this had risen to 200,000 cases of sepsis. And currently in the UK, sepsis claims more lives than lung cancer and more than the combined effects of bowel cancer, breast cancer, and prostate cancer. So this is really is a huge problem. And fortunately in the UK, we have a really excellent surveillance program that's constantly surveying the key pathogens linked to the National Health Service. So all hospitals have to send especially E. coli and Staphylococcus aureus isolates. So every bacteremia case in the United Kingdom we know about. And so we've experienced this 17% increase between 2015 and 19, and also a 32% increase in antibiotic resistance. And that's summarized with the exact figures in this slide. So we're seeing an increase in sepsis in general and an increase in the resistance associated with these cases. Okay, so if we look at the pathogens which are responsible for sepsis in the UK, we immediately see that our huge problem is with E. coli, which is very much more so than the next 7 pathogens combined. So in the United Kingdom, over 50% of sepsis cases are caused by E. coli. And this rate has been increasing. So you can see the figures here from 2015, 2016, 17 to 19. Can you actually see me moving the mouse marker?
Yeah.
You're good. So sorry, it's very, very quiet at the other end, of course. I was just hoping that my internet connection wasn't a problem. So if we go on, if we actually look at the E. coli themselves, we also see that the burden, the antibiotic resistance burden from E. coli shown with these vertical shading in red has been increasing through the years. And it's more important than all these pathogens combined. So probably a different situation in the UK to India, but E. coli is our main pathogen, and by far. And the question is, of course, what on earth is causing these increasing sepsis rates? So in terms of E. coli, I'm of course talking about extraintestinal pathogenic E. coli infections, XPEC infections, not talking at all about intestinal infections such as caused by Shigella dysentery and so like. So I'm talking about urinary tract infections. E. coli in the UK causes 80% of our urinary tract infections, over 50% of bacteremias. It's also the leading cause of neonatal meningitis and various other diseases. And what's really fascinating about E. coli is it's an incredibly diverse species, so that it's possible that if by taking 2 E. colis and comparing them, it's theoretically possible that they could be as much as 80% different. So this is huge. This is like my research assistant Jordan compares this to comparing humans with bananas. Amongst E. coli, they can be hugely different. And of course, we need a classification system to classify these. And traditionally, this has been done by phyla grouping, easily done by a multiplex PCR reaction. You can look at any E. coli and put it into one of about 5 boxes, A, B1, B2, phyla group C or phyla group D. And the interesting thing here is the pathogens mostly belong to this phyla group, the B2 phyla group, but also to over here somewhere, the D phyla group. And we can also go very much more exact by doing multilocus sequence typing. And E. coli now can be split into more than 10,000 sequence types. And the striking fact here is that only 0.06% of these sequence types are responsible for more than 60% of our cases of sepsis associated with E. coli in the UK. So if we could just remove these 5 or 6 sequence types, we would reduce sepsis in the United Kingdom by 60%. And where are these actually coming from? Well, they're coming from us. They're coming from our own gut. Most E. coli infections, urinary tract infections, are seeded from our own guts. So this is very much a carriage problem in the United Kingdom. So another thing about the surveillance in the United Kingdom is we know that the E. coli bacteremia rates in the UK are regional, and they can be very different depending on where you live in the UK. So for example, in London, the rates are very low, and they're very much higher in Wales. I actually live down here at the bottom of Wales, and they're also high in Northern England. They're high in Northern Ireland, and not shown here, but they're particularly high in Scotland. And these rates have been changing through time, as I've mentioned before. But in all cases, the bacteremia rate in England is considerably lower than the rate in Wales or the rate in Scotland. So one of the plausible explanations to this is that we have an aging population, rapidly aging population, and because there are more susceptible people in our population, then the rates are linked to our aging people who are more vulnerable to E. coli sepsis. However, the surveillance has also showed that that certainly isn't the case, because the sepsis rates have been increasing for males and females in virtually every age group. So it's not due to our aging population, it's due to some other factors. And I've been, of course, interested in E. coli for several years. Here's a study I think I showed you last year, so I'll just mention it briefly, in Pakistan. And the take-home message from this study, looking at carriage of E. coli in Islamabad in Pakistan, is the carriage rate of these pathogens was less than 1%, very low in Pakistan. And then when we looked in Silchar, a city in northern India, although there was huge amounts of resistance carried by the population in this city, notably NDM5, actually when you looked at whether these E. coli are pathogenic or not, the bottom line is we found 0% of pathogens. So huge problem with resistance in South Asia, but not a huge problem with pathogenicity associated with E. coli, which is really very interesting. So we wanted to know about the UK. So we did a study with 6 sewage works in the United Kingdom. And the very important thing with sewage coming into sewage works in the United Kingdom is it's heavily mixed. It's homogenized. So as you see it actually coming into the sewage works, I thought when I went the first time you'd see lots of brown things floating, and it's just not true. It's absolutely homogenized, and this is fantastically useful, because basically what's going on is from one sewage from one sewage sample, you can work out the carriage rates of a whole city. So maybe 1 million inhabitants in, for example, Bristol, by sampling one single sample of sewage, you can work out the carriage rate of different types of E. coli for 1 million people. So there's a motorway linking London to South Wales, which runs along here. So we chose 3 sites within 40 miles of London and 3 sites within 40 miles of Cardiff, where we work. And we collected these just before we came to the last Bacteriophage conference. And this is our methodology. We dilute the sewage roughly 2,000 times more if needed. Plate it on UTI Brilliance agar, which gives us a color indication for possible E. coli's. We dilute it hugely so that we only get 5 or 10 red colonies a plate. We unbias any picking of the E. coli by anything which is pink or anything slightly red color. We pick everything. So we pick all the red colonies. We then do multiplex phylotype PCR. We confirm the species by MALDI-TOF, and we sequenced all the pathogenic types. So out of this, from these various sewage works, we recovered 98, 160, 112 E. coli from each site. And we also sampled Dhaka at roughly the same time, about a month later. October 28, we took 9 samples from around Dhaka in Bangladesh. And then we used one site, which is very close to us, Newport, to confirm the accuracy of our methodology. So these are the results, and they're really quite interesting. I really want to focus just here on the true pathogens, the B2s. And essentially in the Western sites, the carriage rate was huge, getting on towards 30%. So 1 in 3 people at least carrying these pathogenic E. coli types, compared to 0, close to 0% in South Asia, clear difference. So in the UK overall, 21% carriage compared to 0 in Bangladesh, and a clear difference between the sites in the West and the East. So if I put this on a map, here's the sepsis rates, the high sepsis rates here in Wales. The diameters of these pie charts are showing you the percentage of the pathogenic E. coli. So Bristol 23%, Newport 33%, Cardiff 26%, very high. And again here on the close to London, you're seeing a very much lower carriage of pathogenic E. coli types. And here's just some of the statistics on this sampling. So this is the Newport site. Here's three selections of around about 100 E. coli from the same sample. And we do the statistics on this. The p-values are very low, telling us that these are really different between the different phylotypes. And yet the p-value here is 0.6 for all this group, showing us that there's no difference between our analysis here between these 2 different samples. So one taken in September and one taken in October, and one sampled 3 times and the other twice. And then it's also statistically relevant for East versus West, p-value of 9 times 10 to the minus 5. And it's also statistically relevant between England and Wales. And then, of course, we've sequenced these. So, and here's some really interesting information, and that is the most prevalent of these pathogenic phylotypes are the very same ones which are causing the sepsis events in the community and also in the hospital. So our problem with sepsis, our huge problem in the United Kingdom with sepsis, is directly linked to the carriage of just these handful, really, of sequence types of E. coli. And of course, the fact that there's difference between South Asia and the United Kingdom means that it can naturally change. And so here is an analysis of these same sequence types and the amounts of the prevalence of these between the near London in yellow and the ones in Wales in blue. And so the difference between the carriage rates in the gut carriage rates of these pathogenic strains in Wales is considerably higher to those close to London, which was absolutely reflecting these sepsis rates. So our sepsis problem is a carriage problem. It's simply a carriage problem. And so if we reduce the carriage of these pathogenic types in the gut, if we target these with bacteriophages, orally applied bacteriophages, then I believe it's highly likely that we can reduce the carriage rate of these and reduced infections themselves. So I just very briefly show this. I think this is my final slide here. So this is a study we've done on the most prevalent of these sequence types 131. I think Jordan showed it earlier on in the week in his talk. But basically, we've looked at different nations, and we find that the nations with a higher prevalence of gut carriage of these resistant pathogens have the lowest number of big bacteriophages. So each of these numbers here is a separate sequence type 131 strain, and we used about 200 different sequence type 131 strains collected from all over the UK, from sewage, from bacteremias, from fecal samples, and we searched for bacteriophages in sewage, and we see in South Asia you've actually got huge numbers of bacteriophages killing this strain, which is probably why you have very low carriage in South Asia of this particular strain at the moment. Although we know that 20 years ago, this was a huge problem actually in India. So it's a problem which has vanished over the last 20 years, and but is increasing in the UK. So in summary, E. coli bacteremia rates are directly linked to high carriage rates of pathogenic E. coli types in the UK community, notably just the small number of sequence types. E. coli in South Asia are generally of different types to the UK, with very low carriage of pathogens, but high rates of antibiotic resistance. This is really interesting. The UK has a huge problem with carriage of virulent E. coli, and South Asia has a huge problem with carriage of resistant E. coli, but not pathogenic. So you have to ask the question, which is the worst problem, carrying virulent ones or carrying antibiotic resistant ones? I think it's probably the carriage of the virulent ones. So the rising rates of E. coli sepsis in the UK likely reflect the rising carriage rate of these pathogenic types through time. This is very similar in Europe and also in North America. And I've also shown you some information that suggests that bacteriophage are involved in this natural process. And since it's a natural process, it is possible to manipulate this process by adding bacteriophages to the guts of people in the UK. So I'll leave it there. Just to acknowledge people in my group working on this, so especially Jordan Matthias, who gave a talk earlier in the week, Abdul Rahman, a PhD student from Saudi Arabia, and Dr. Willems, who's a postdoc working with me from Brazil. Thank you very much for your attention, and thank you so much for inviting me and for a really interesting few days. Happy to take any questions that anybody may have.
Excellent presentation, Mark. Thank you for accepting our invitation.
Thank you.
E. coli is a very serious problem, not only in UK, but even in India. Majority of the E. coli, even what we are isolating in our surrounding or in the environment, are extensively drug resistance. And the majority of the STP, in spite of a very efficient sewage treatment strategies, E. coli is escaping. So we need to use a higher concentration of the disinfectant. For that matter, it could be chlorine, but we are unable to do that. And we have tried to prove that the phages are more effective in place of chlorine. Thank you for a wonderful presentation, Mark, and it's open for discussion.
Thank you very much, Mark. I am Gopalnath from Varanasi, and it has given a new insight to me, especially personally, because we are working with the gut microbiota and the obesity and the non-alcoholic fatty liver disease. So we are trying to pinpoint the E. coli strain because you are telling that there are more than 10,000 ST types. So if we are getting something related to obesity or diabetes or non-alcoholic fatty liver disease, with bacteriophage therapy, we can eradicate and we can get the result. So this is great. And I am personally going for the sequence typing of the E. coli, whatever we are getting from the obese patient and also from the diabetic patients. And this non-alcoholic fatty liver disease is basically, the etiology is not known. So this may give the new insight. Maybe we hope that something is happening, but this came in my mind because of your excellent lecture. Thank you very much, Mark.
Thank you so much, Opal. Thank you. It's very interesting. I don't know whether you've read the papers, but if you look at the carriage of E. coli in individuals, the majority of people, 60%, only carry one single sequence type. So, and another 30% are carrying two sequence types. So we've also looked in the lab at what we're carrying ourselves. So, and most of us are carrying pathogenic types. So it's of a special interest to us.
Sure. We'll be looking into it. This will be very interesting to see with the obese patients, what is happening with them, and then compare with the skinny people, that what is happening in them, what sequence type is there. Okay, this was great because
Thank you.
We have used the bacteriophage orally in the animal model to eradicate the infected bacteria. So we have already done published it.
Fantastic. Thank you.
Thank you, Mark. It was a fantastic presentation and work. Thanks for your time.
Thank you, Ramesh. It's a pleasure.
Thank you.
Dr. Mark, Prashant here. I have a question for you. Yeah, how broad your bacteriophages that were isolated in UK? Do you have bacteriophages with broad host range?
Yeah, we've been testing the host range. We've got something like at least 300 phages against sequence type 131. So it's too many to do the host range experiment on. So we've kind of chosen the 30 best ones, and we've only so far looked at other pathogenic types, and they seem to target mostly the sequence type 131 ones. Type of bacteriophage. What we haven't done yet is we haven't looked to see if they target any of the commensal types as well. And of course, that will be very important for their use as trying to reduce the pathogenic types. So it's in process, but as you know yourself, it's an awful lot of work working with hundreds of bacterial strains.
Great. Thank you, Mark. Thank you for joining us today.
Mark, whether all these E. coli which you isolated are from the human origin?
So basically, we've got a collection of around about 300 sequence type 131 isolates, which were collected in 2014 from a large UK study. So it includes bacteremia isolates from hospitals, 5 regions in the UK, sewage isolates, 1 or 2 isolates from animals, very few, but and also what's the other one? Farm animals. So it's kind of like a One Health collection, really, of every area we could find sequence type 131.
We are isolating the E. coli which are drug resistance, and we have not sequenced it. So if you like to have a collaboration, I can definitely ship for a good number of E. coli to your lab, where you can do a sequencing study and you can use a fast typing to identify them.
Sure, we'd be very happy to be involved with that. Indeed.
Thank you, Mark. Thank you very much. If there are no questions, Mark, thank you once again. And we appreciate your constant association with our society. As you rightly mentioned, a couple of years back, we all met. The other day, Sebastian was showing the Phage T-shirt given by Ramesh. I was thinking that you two would be wearing that. But anyway, thank you, Mark. Thank you very much once again.
T-shirt given by society, Dr. Vinod sir. I'm not.
It's your idea, Ramesh, that the society wants to give credit to you. I think the Gopalnath sir will definitely agree with that.
Sure.
If there are no more questions, with the permission of the chairs, shall we proceed to the valedictory function? Because it was posted for 3:30. I think we can advance it now and we can continue with that.
Yeah.
Yes, sir. Yes, we can.
Thank you very much. Dr. Sanjay, sir, are you there? Dr. Sanjay? Dr. Sanjay?
No, sir, I think he's not here.
He's registered. So he's there, but I don't know why.
I'll call him, and we'll start in a couple of minutes. We'll start the valedictory in a couple of minutes. Thank you.
Thank you.
So an important announcement. Following this valedictory, there is an annual body meeting of SBRT. This is only for the life members of SBRT. Others should not be. Thank you.
Indian, there are 2 questions in the chat box. Maybe you can ask them.
To Mark. There are 2 general questions. I think even Professor Gopalnath can also answer. They are saying the first question is: does phage therapy, oral administration, also has to undergo the same clinical trials as any novel antibiotic?
Yes, definitely.
I believe in the I believe in the UK it does, unfortunately, and I don't really understand why, because you can eat bacteriophage treated meats and things, so it seems ridiculous to me. I quite happily swallow a flask full of them and try and take out my pathogenic E. coli types.
So that question was by Shafa C. Okay. So next question, I think the name is not there, the phone name is there. So will the bacteriophages also affect the normal E. coli in the intestine?
Yeah, so it of course depends on the host range. So we're doing work to make sure that we only use ones which are targeting pathogenic types, and hopefully that will not change the microbiome very much at all and allow the commensal types to take over. That's our hope. Very similar to what is what is present in E. coli. You don't seem to have what's present in South Asia. You don't seem to be carrying very many virulent types. So I'd imagine that overall, probably your urinary tract infection rates in India due to E. coli are lower than Klebsiella, for example. Whereas they're very high in the UK. 80% of our urinary tract infections are E. coli. But I don't know. I don't know if anybody has figures on that for South Asia, India, or Bangladesh or Pakistan.
Prashant, you have the answer for this, because I think similar question was asked in your PhD Viva.
Yeah.
So will the bacteriophages also affect the normal E. coli in the intestine? So you have any answer?
Yeah, it does. It does affect the normal flora. We have to be very careful about the bacteriophages, how broad the bacteriophages what we administer. Yeah, we have to be very careful about what bacteriophages we are using for the treatment.
Okay.
I just want to add a point here, and I want Mark to comment on this, because this is a very difficult question to answer, whether the phage will affect the normal commensals, and until and unless the complete characterization of the commensals are not done.
Exactly, that's true, and of course everybody's looking at the resistant E. coli, but there's so very few people looking at the non-resistant ones, and most of these virulent ones I'm talking about are not resistant to antibiotics. So they're termed almost as an interesting. And then again, of course, the commensal ones are termed as even less interesting. So it's even more difficult to work on those. So we're certainly sequencing at the moment a lot of commensal types to understand what are the prevalent commensal ones in the United Kingdom, so we don't actually remove those.
Thank you for that the opinion. And I think Dr. Gopalnath will add a point and agree with me that, the antibiotic-induced diarrhea are mainly because of complete eradications of the E. coli in the gut.
Yeah.
So it is something similar also. We need to have the application of phages here. When you use a phage for anything, for GIT related, you have to be very clear whether it will eradicate the normal flora of the gut. So that correlation is a very important aspect. What is your opinion on this, Dr. Gopalnath, sir?
Actually, what I feel is that if the phages are broad spectrum and are able to kill many other hosts, then they will be acting. So for this purpose, especially in the gut flora eradication, I think very narrow spectrum phages should be picked up, and they will be not disturbing the normal gut flora. So commensal will be. So for picking up, somewhere we are picking up the broad spectrum bacteriophages, and somewhere we have to go for the narrow spectrum. So the pathogens which are being picked up like ST type, those are we having the specific phages for ST131 only? That is the thing which we have to go for this. So that will help us.
Thank you, sir. What's your opinion, Mark? Do you agree with us?
Yeah. So, sorry, were you asking me?
Yes, Mark.
Yes, I agree. We ideally need narrow host range bacteriophage. I think that a lot of the sequence type 131s we have are in fact narrow host range. They're certainly not targeting many other sequence types which we know are pathogenic. So I'm presuming at this point that then they're not targeting the commensals, but of course, until we test them, we don't know that. So that's a priority at the moment.
Actually, in biological system, nothing is absolute. So that is the truth. In medicine, we are taught like this.
Yeah.
Nothing is absolute. Yeah.
Thank you.
Yeah.
Thank you, Mark. Thank you for the excellent presentation and also discussing and answering the important questions raised by the delegates. Thank you. I think Professor Sanjay Chibber is available. Shall we proceed with the valedictory function, sir?
Yes, sure. I'm sorry for being late.
Thank you, sir. Thank you very much. And good afternoon. I welcome one and all for the valedictory function. It's a privilege to welcome all the delegates and office bearers of the Society of Bacteriophage Therapy, Research and Therapy. I welcome President of the SBRT, Professor Sanjay Chibber, Secretary, Professor Gopalnath, esteemed members of the Society, Editor, Dr. Ramesh, and the invited speakers of the conference, chairpersons, moderators, and above all, the delegates. As the convener of the conference, I thank the Society for giving us the opportunity and the responsibility to conduct the second international conference on bacteriophage research in the virtual mode. On record, around 120 delegates participated in the conference and 60 delegates presented the research work by oral mode. The delegates were from different states, almost 9 to 10 states, namely Chandigarh, New Delhi, Haryana, Madhya Pradesh, Uttar Pradesh, Andhra Pradesh, Karnataka, Tamil Nadu, Maharashtra, Kerala, and around 17 speakers. They deliberated over the period of 2 days, and we had international speakers from United States of America. We had 2 speakers from United Kingdom, and we had a speaker from Spain, Turkey, and Germany. And also we had one more speaker, and is among us. That's mainly is from China, and he's working in China. And the Indian speakers were from Punjab University, Dr. Professor Sanjay Chibber, Gopalnath sir from Varanasi, and we had a speaker from Vellore. We had a speaker from Madhya Pradesh, Andhra Pradesh, Kerala, and also Karnataka. I am really overwhelmed with the support I received for the conference. In spite of all the educational institute were closed, students showed a lot of enthusiasm in the preparation of the abstract and making a presentation in this conference. This shows, Professor Sanjay Chibber, please mute your mobile. Yeah, let me mute your microphone. This shows the interest in the fast therapy. And once again, I thank one and all for showing a tremendous support in organizing this conference. And I request President Sanjay Chibber to speak few words.
Dear delegates, participants, ladies and gentlemen, the 3-day virtual conference, which was held by Dr. Vinod Kumar and his team, has been very successful. We really enjoyed the scientific deliberations on all these 3 days, and the enthusiasm shown by the young researchers was excellent. I appreciate, and I feel they should be encouraged in future also, because in fact, this is the only platform for them where they can come and showcase their research, and they can also interact with the senior scientists within the country. So I am very happy that enthusiasm was there among the young researchers. The second day of the conference, yes, there were invited speakers, both from India and outside India, and there were good lectures. In fact, we all enjoyed listening to all these lectures, and they were very, very informative and very, very useful, not only for the young researchers, but even for scientists like me. We have learned a lot, and I am happy the third day also saw lectures from the faculty, and it was really good, and we really are thankful to the organizers for having selected such good speakers who not only gave the like information about bacteriophages and their future, but it was also very meaningful. And therefore, I congratulate Dr. Vinod Kumar and the team. And the only message I would like to give today is that we are a growing society, and we would like that more and more people, when I say people, I mean the scientists who are working in the area of bacteriophages, should come and join the society. And because it is an upcoming field where bacteriophages are finding a lot of application. In the future, this will be a very meaningful source of many in many different areas of the science, and therefore it is going to have lots of applications, lots of interest, and we all should get together, discuss all the things, and take it further. So with this, I once again congratulate Dr. Vinod Kumar and his team for doing this conference in such a nice manner. And I'm also thankful to all the members of the Society for Bacteriophage Research and Therapy who have participated also with lots of enthusiasm in all those 3 days. So I wish all the best and all the good luck to all of you. Thank you.
Thank you, sir. Thank you. Thank you very much for encouraging words. So I request Secretary Dr. Gopalnath to speak few words.
All dear delegates, the organizers, the speakers, and the whosoever participated or actually helped in this conference. I have to say only one sentence, that it was simply overwhelming 3 days. On the very first day, the deliberations, the presentations by the young scientists was really so nice that I could not leave my computer. So I was just jumping from this session to that session to that session. So that was really very interesting. And this virtual mode conference organized by Professor Vinod Kumar, I know that he must have worked hard for 4 or 5 months. And I really congratulate, because this third day is really very satisfying. It is giving me immense pleasure and satisfaction, I should say. And I would like to remind that the journey which we started from Varanasi on August 22nd, 23rd in 2017 is just today we are sitting here on the day of July 23rd, 24, 2021. So this is great. Only the problem, Dr. Ramesh could continue in the 2019 very well after the registration of the society. And a great conference was organized by him in the name of International Conference on Bacteriophage Research and Antimicrobial Resistance under the umbrella of SBRT at VIT Vellore. That experience was really fantastic. So I have mentioned again that we really enjoyed Dr. Ramesh at that time. And here this virtual conference really was very interesting. And this has given a lot of lesson to us. So in future also, so many foreign speakers participated. We can have that one like in this mode only in coming days if they are unable to come. So it was only COVID-19 which halted somewhat, but we are not discouraged. And I'm tempted to this point to state that we sowed the seed of Bacteriophage Society altogether, and it is great pleasure watching that this society is growing and thriving very well under the this group altogether which we are working. And we can feel and we can sense that everybody is doing it wholehearted for this society. So this is very satisfying and encouraging both. So I hope that this scientific feast will induce the students, scientists, and clinicians working in bacteriophage research to contribute with their full potentials to fulfill the need of the country in all the sectors of the life sciences. And it was really interesting to hear from all the basic scientists to the application scientists in bacteriophage therapy from Indian side and also from outside. So this was a great experience. And again, I have no word to express my feelings about Dr. Vinod, what he has done for us, for this society, although this was his duty to do this. So I'm again thankful to him, congratulate to him. And then I sincerely thank and wish all the best of luck to all the members of the society whoever participated for the future endeavors. Here I would like to tell that somebody must volunteer for the coming or next conference by himself without our request. So it will be great for us. With these words, again, I thank to all of the members of the SBRT and all the speakers and delegates whoever participated and whoever helped us in organizing this conference. With these words, thank you again and best of luck for this SBRT. Thank you very much.
Thank you, Gopalnath sir, for your appreciation and encouragement. Thank you, sir. Thank you for your support, because it is not just we know that, but you rightly said it's the teamwork. It is right from the the core committee members. Maybe Sanjay Chibber, Gopalnath, Ramesh, Urmi, Sangeeta, Taruna, Ajay, all have contributed. From past 4 months. If at all it's a success, it is a success of the society and it is a success of the bacteriophage. Thank you, sir. Thank you very much. And again, with the kind permission, I would like to move to the next stage, that is the award ceremony. I'm going to share the slides. Is the slide visible, sir?
Yeah. Visible, Dr. Vinod.
Yes, thank you. The core committee and the members of the SBRT, it was decided to honor the scientists who are working on the phages. So it was unanimously decided by the society to give away these awards: Lifetime Achievement Award, Outstanding Scientist Award, Young Scientist Award, and the Best Oral Presentation Award. And it was also decided initially, I think Dr. Taruna will agree, It was even decided to add a poster presentation, but we received only one poster for the presentation. Hence, we requested the presenter, to convert it to oral presentation. So we have only the best oral presentation. But on recommendation from the president and the secretary, it was decided to give 6 awards in the best oral category. And also it was again decided by the committee to use a different phage scientists who have contributed and dedicated their lifetime, on working on the phages. It was decided to name a lifetime achievement award category in the name of Ernest Hanbury Hankin, and the Outstanding Scientist Award in the honor of Felix de Herel, and the Young Scientist Award in the name of Elizabeth Martin Cutter. Why the Ernest Hanbury Hankin? Just for those who are interested, Ernest Hanbury Hankin was an English bacteriologist and also the aeronautical theorist and the naturalist. He was working mainly in India, and he was studying mainly in the field of malaria and the cholera and other communicable diseases. He is often considered as among the first to detect bacteriophage activity, and suggested that their presence in waters of Ganga and the Yamuna River may have led or may have a role in restricting the outbreaks of cholera. And Felix de Herel, I think majority of you know that, he was a person who co-discovered the bacteriophage, and he dedicated, and also, he was executed, in fact, for working on bacteriophage. And he was in exile for many years. He worked on Shigella and many of other communicable disease also. Similarly, it is named, the Young Scientist Award is named after Elizabeth, because she's in USA, and she is a great contributor, and she's a director of Evergreen Conference and the Society. She has dedicated almost from 40 years, she is working on the phages, and she was a invitee for the conference which was held at Vello. Coming to the first one, that's the Ernest Hanbury Hankin Lifetime Achievement Award. This category was directly selected by the society itself. We didn't invite any nomination for this award, and the society unanimously decided to give away 2 Lifetime Achievement Awards after Ernest Hanbury Hankin. The first award goes to Dr. Sanjay Chibbar. I request all the members to clap for this. Please, you can unmute. And Dr. Sanjay Chibbar or Professor Sanjay Chibbar, yes, thank you. Thank you very much. Dr. Sanjay Chibbar, he did his MSc and PhD, and he has a total of 42 years of experience, and out of which 20 years, he has been working on the bacteriophage. And he has around 51 publications in the field of phage, and he has handled around 4 bacteriophage projects, and he has 3 patents in his name, and he has given around 22 talks only on phage, and stressing only on phage. So there are 51 publications. It's a huge contribution by this person. Is always encouraging the youngsters, and I don't know, he has not mentioned about how many PhDs he has awarded, in the field of phages. So it is a great honor to award this, or to give away this award to Dr. Sanjay Chibbar. Yeah, this is a certificate for Dr. Sanjay Chibbar for his immense contribution in the field of bacteriophage research over the period of 2 decades. Now I request Dr. Sanjay Chibbar. Are you there, sir?
Yes, I am. Yes, thank you so much, Dr. Vinod. Thank you.
You can speak 1 or 2 words about your opinion about this awards and also the society.
Yeah, I am thankful to the society, the members of the selection panel for bestowing this award on me. Yes, I have worked in this area for many years. In fact, when not many people on the international arena were also interested in bacteriophages. So we started working at that time at Punjab University. And over the years, we have taken it from the scratch in my laboratory to a level where today we can say that we have a product which can be used for the welfare of the human beings. So I feel very proud and honored that the society has bestowed this award. And I thank once again to all the members, and I would encourage all the young scientists also that keep on doing your work. Never look for any awards and things like this. They come to you on its own. And thank you very much.
Dr. Sanjay Chibba, thank you. The next Lifetime Achievement Award goes to Dr. Gopal Nath. I think he is the person who deserves this award from past 18 years. Yeah.
Thank you very much.
Dr. Gopal Nath has done his MD and PhD in microbiology, and he has over 36 years of experience, out of which 18 years, from last 18 years, he has been working on the phages. He has around 18 publications, handled around 6 projects, and he has 1 patent, and he has given around 15 talks on phages. And he has handled a lot of clinical trials on phages. So on compassionate ground, this is very important, and the youngsters has to understand, on compassionate ground, you can use phages on human being, provided you have a permission of the treating physician. I think the Gopal Nath sir has been doing this for many years. He has published excellent papers. It is not just a publication in a predator journals. He has published papers in a very good journals. So it is an honor for the society to bestow this award to Dr. Gopal Nath sir. Gopal Nath sir for his immense contribution in the field of bacteriophage research over the period of 2 decades. I request Gopal Nath to speak few words.
Thank you very much. This is really unexpected achievement to me. When I started working in this area, I was never thinking that one day I shall be reaching to this level, that going to get the Lifetime Achievement Award. I was simply working that I have to work in this area because this is new and having very potential for the human health or the whole living being. So I don't have the words to express. And then again, we organized this and we made this society, not thinking that this will be happening. So this is Dr. Vinod, and Dr. Chibbar. I am so thankful to you that I never expected. So I don't know that I was never thinking that I shall be getting a Lifetime Achievement Award by any of the society. So I shall keep on working in this area. And right now I have made the atmosphere in the clinical colleague also. They are coming up. We are discussing and we are doing. And I request to all the youngsters that they can think in application part of the bacteriophage research and therapy. That will be the great contribution. And in coming years, one day we will be able to convince the policy makers that they will be permitting for the clinical use fully. And on compassionate ground, actually we are using, but not publishing, because these are the individual cases. So whosoever requests us, we have treated so many cases of the chronic osteomyelitis and also few cases of septicemia, but we have not published. So we are doing this, Dr. Vinod, and Dr. Sangeeta is there. It's so nice to see you all here. We all are the team members, family members playing all together. And one day we'll definitely succeed and make a lot of goals of the winning end. Thank you very much to SBRT and to Dr.
Thank you, Dr. Gopal Nath. And we eagerly look forward for your guidance and for the collaborative works. Thank you, sir.
Welcome.
Next award is the Felix D. Herrell Outstanding Scientist Award. The society decided to give away the 2 awards. I request Gopal Nath to continue the award ceremony. Dr. Gopalnath, are you there?
Yes, I'm here. So change the slide. So society has decided to give away this award, Felix D. Harrell Outstanding Scientist Award 2021 to Dr. N. Ramesh. And we know his contribution is immense, and he is very active member of the society, and his academic qualification is PhD. Total year of experience in the phase area is 15 years. And he has published 17 papers, and number of projects he has brought 1, and number of patents he has published 1, and he has delivered about 32 talks on phase related one. So this is I congratulate to Dr. N. Ramesh, and I hope that he'll be coming up in a great way with the journals and other publications which we are intending to bring in future. And again, this is the this outstanding award. We are so happy to give away to Dr. Ramesh, who has contributed immensely to our society. So congratulations once again, Dr. Ramesh.
Thank you, sir. Thank you so much, sir. Thank you so much for the great award. It's my great my great honor to receive this award from the society. Still, I have a lot of responsibilities to serve our society and throughout the world. We have a lot of job to do that. Definitely we'll do, sir. With your great support and blessings, we'll do a lot, sir. Thank you, sir. Thanks for the complete society's official bearers. Thank you so much, sir.
You are welcome, and then you are the flag bearers of the society in coming years. So that's why we are just trying to give the load on your shoulders. So you have already taken. So in future also you'll be doing this. So again, once again, congratulations to you.
Thank you.
Dr. Binod.
Yes, sir. Next slide.
So the next Outstanding Scientist Award goes to no one else than the Dr. Vinod Kumar. Actually, I would say before going to all his credentials that I read his paper on Pseudomonas aeruginosa septicemia, and I think you published in 2007 or 9 something.
Yes, sir.
And from there, I took the materials and method part, and then I started my work. When I went to the murine model experiments. So that was the great contribution to my work. So you may not be knowing, but I have to mention here. And you definitely deserve this Outstanding Scientist Award, Dr. Vinod, and the society has endowed on you. So this is the great thing, and it is not only honor to you, we are feeling honored by giving this Outstanding Scientist Award to you. So his qualification is PhD Bacteriology, PhD Virology. So he had done two PhDs, and total experience on bacteriophage therapy research or bacteriophage research is 21 years. And then he has published 132 papers. Total number of publication in phase area is 25. Number of projects he has handled 4, and number of patents he has got 1. And he has delivered almost 20 talks at different platforms. So congratulations once again to Dr. Vinod Kumar, and I hope that you will take this society to the great heights.
Thank you very much, sir. Thank you very much. It's a great honor to receive award from the society. I was a Young Scientist Award awardee, receiving the award from President Abdul Kalam sir, and I'm equally overwhelmed to receive this award from the society. Thank you very much, and thanks for your constant guidance and the support, sir. Thank you very much.
Welcome, Dr.
Thank you, sir. And the next category of award is the Elizabeth Martin Kutter Young Scientist Award 2021. We received around 18 nominations for this award. It was really tough for the judges to evaluate and to give away the 3 awards. It was very difficult time to scrutinizing, because I've been always in touch with the president, Sanjay Chibber, or the secretary, to how to go about. A lot of factors criteria were included, deciding who deserves, because the main idea is we want to encourage the young scientists, because if at all, if the Phage should be a success, it is mainly by the young scientist. So And the first award, it goes to Dr. Prashant Manohar. Manohar has done his PhD. He has a total years of experience, around 5 to 10 years. That's what he has written in his nomination. And again, the phage therapy experience is 5 to 10. He has a very good number of publications, very good in a very high impact factor journal, around 15. And he has a patent. He has published a patent is one. And he is presently working in China. So congratulations, Prashant. Are you there? Prashant?
Yeah, he is there. He is there.
Is there? Prashant,
Yes, sir, I'm here, yeah.
Congratulations, Prashant.
Thank you so much, sir. Thank you so much.
Scientist Award 2021.
Thank you so much, sir. So wonderful pleasure to receive award from the society. I strongly believe that this award encourages me to do more on bacteriophages, and I think I will come up with something new in our next conferences. Thank you so much. Thank you so much, all the society members and judges for choosing me for this award. Thank you so much, sir.
Thank you, Prashant, and congratulations once again. All the best. And the next Young Scientist Award goes to Dr. Sangeeta Aiwale. Sangeeta, are you there?
Thank you, sir. I'm there, sir. Thank you very much, sir. Thanks a lot.
Qualification. She has done a PhD in microbiology. She has around 15 to 20 years, and she has a publication, 12 publication in her credit. And out of 12 publication, 11 publications, she is a first author. That is one of the criteria which was considered in 12 publication. I repeat, she is the first author in 11 publication. Congratulations, Dr. Sangeeta, once again.
Thank you, sir. Thank you very much, sir. So I'm very much thankful to the society, and in future also I will work hard and I will be there for the society, and the Bacteriophage consultancy which I am giving from last 10 years, I will continue that also, sir. Thank you very much, sir.
All the best, Dr. Sangeeta. The third award goes to Dr. Amita Gupta. Dr. Amita Gupta, are you there? Dr. Amita? Yeah. So Dr. Amita, she has done PhD in biochemistry, and she has around 15 to 20 years of experience, and she has published around 7 papers, and she has 4 patents in her name. And it's a great honor to recognize your contribution, Dr. Amita Gupta. Congratulations. And okay, maybe your slide.
Thank you, sir. Thank you so much. I am really grateful to the society that they considered me worthy of this coveted award. And I look forward to working with the August gathering, the members of the society, to take phage display technology and phage therapy further, so that it can benefit the society. Thank you so much, sir.
Thank you. Congratulations, Dr. Amita. Best Oral Presentation Awards. It was, I think, 24 hours back. It was decided by the president and the secretary of the society to give away 2 awards from each category. There were totally 3 categories. 60 students, they made an oral presentation. From the hall 1, that is the category 1, the award goes to Rutuja Gangadkar. The title is a molecular dissection of N-terminal catalytic domain on D29 phage peptidoglycan hydrolase. Address ISCR Bhopal. Rutuja, are you there? Rutuja?
Yes, sir.
Rutuja,
yes,
congratulations, sir. Rutuja.
Congratulations.
Thank you, sir. Thank you.
Congrats.
Thank you, sir.
Congratulations.
Thank you so much. Thank you, ma'am.
Yeah. Next award goes to Priya Moria. Priya Moria, and the title is Bacteriophage-based biosensor for the early detection of fecally contaminated water. Mahatma Phule Mahavidyalaya, Pune. Are you there, Priya? Priya, congratulations.
Priya is not there, sir. Actually, she's not.
Convey the award, madam.
Yes, sir.
Convey
Yes,
the award.
sure, sir.
congratulations for her team.
Yes.
And from the hall to the 2 awards, there is Rajita Satish. The title is Isolation of Mycobacterial Phage, a novel tool to treat mycobacterium. She's from Department of Microbiology, SIES College of Arts and Science Commerce, Mumbai University, Mumbai. Rajita Satish?
Yes, sir. I'm here. Thank you, sir.
Congratulations,
and thank
Satish.
you, society. Yeah, thank you, sir, very much.
Thank you and
It was
congratulations.
a wonderful experience interacting with everyone. And even I got so many ideas and inspired by the interaction that happened. Thank you very much.
Thank you. That is the very purpose of such conference. And I hope you are given the positive feedback. Thank you very much, Rajita. The next goes to Archana L. Title is Antiphaging and Fitness Trade-off During Phage Bacterial Co-Evolution in Staphylococcus aureus. From Hall 2, Vellore Institute of Technology, Vellore. Archana, are you there?
I think you're there. I think no, sir. So.
Hello.
Archana? No, she's not there. Anyway,
Yeah.
please convey the award.
Sure.
And I congratulate Archana and her team, for getting this Best Oral Presentation Award. And the last one goes to Virendra Bahadur Yadav for the in vitro treatment of Salmonella typhi infected peripheral blood mononuclear cell using specific bacteriophage. That's in Hall 3. Address Department of Microbiology, Institute of Medical Sciences, Banaras, Hindu University, Varanasi. Congratulations, Virendra Bahadur Yadav. Virendra, are you there?
Yes, sir. Thank you, sir. Thank you very much, sir.
Thank you, one and all. I request the delegates, 1 or 2 delegates, to speak a few words.
Actually, I feel very nice.
The last one. I missed the last one. That's the 6th one. The 6th praise goes to Dr. William Martin. The title is In Vitro and In Vivo Effectiveness of Bacteriophage Cocktail Against Multi-Drug Resistant Klebsiella Pneumonia. That is Hall 3. And address is the Cardiff University. I think Mark, are you there, Mark? I think Mark.
Yes, I'm here, and I'll accept it on Will's part. He really is a great researcher, and he needs a fantastic young scientist. So thank you so much for that. It's very much appreciated.
Thank you, Mark. Thank you very much. I request participant, 1 or 2 participants to speak few words, please.
I think Rajita is here.
Yeah.
Rajita? Yes.
Thank you. Sir, first of all, I would like to thank everyone in the society. Actually, just 1 week back, July came to know about that event, but I just got the message. I remember messaging Vinod sir because that was a number there. And then sir was so grateful, he immediately sent me the brochure, and that's how I registered. So I would also like to take this opportunity to thank many of the scientists, because when I was doing my work, I have read some of your papers, and actually getting to meet all of them, I can't tell him, I felt so happy. Especially, I would like to thank Urmi ma'am and Indian sir. I don't know whether they remember, like when I was working on Mycobacteriophage and I had a doubt, I had sent message to ma'am, and she was so sweet of her. She immediately replied and connected me to Indian sir. And so many times I had messaged, I am actually mailed him, and he has been always grateful and replied back to all my silly queries as well. So, and even when I came in this, I don't know if whether both of you will remember this thing. And even when, as I told you, like right from Sir Chibra sir to Gopal sir and many others whose resume also I saw, And trust me, I feel so inspired that there are so many things that I can do and which I should start working on is what I felt. So I'm thankful for such an interaction, such a lovely interaction to all the scientists and especially to the team throughout the event. Everything was very well communicated, and there were no issues. Like I didn't face any issues. Even online, there were no issues. I'm very thankful to all of you. Thank you very much.
Thank you, Ranjita.
Yes, I remember, Rajita. Congratulations.
Thank you, sir.
Thank you.
Anyone? Any other delegates want to speak? Other delegates, please.
Good evening, everyone.
Good evening.
Thank you for the award. And I really would like to thank the organizers for this very great and informative conference. Thank you so much.
So, Rutuja, this is Rutuja, right?
Yes, sir, I'm Rutuja.
Your phone name is visible here.
Okay, sorry. Yeah, this is Rutuja.
Thank you, Rutuja. Anyone else, please? Feel free to speak. We have 2 more minutes.
Yeah.
Archana, yeah, okay, Karen, please.
Sir, Virendra here. First of all, thank to all of you for selecting me for award. And first, when I met with Dr. Vinod sir in 2017, he felicitated me very much. So I started to work in the guidance of Professor Gopalnath sir. But always there was a role model for me, Dr. Vinod sir. So today I am very satisfied, sir. Thank you very much, sir.
Thank you, Virendra. All the best. Thank you.
Archana, are you there?
Yes, sir. Yes,
Yeah,
sir.
please. You can share your experience.
Archana, you got a best oral presentation award. Congratulations for that.
Thank you, sir.
You can give your feedback in case you wish to.
Yes, sir. Sure, sir.
PPT is not required for feedback.
Sir, no, sir. So thank you so much. Just now my sir told me that I've got best oral award. Thank you so much. And the conference was really awesome. I enjoyed the first day as a moderator. And the last 2 days I was not able to join due to some lab stuff works. And I really loved having different, yesterday I was going through YouTube live videos, and then on the talk given by so many people, and the keynote was really awesome, and it was really giving me a lot of insights. Thanks for that. Thanks for Dr. Vinod sir for organizing such a wonderful conference. Thank you, sir.
Thank you very much. Anyone else left? Fine, thank you. And at the end, for the courtesy sake, I request our Honorable Secretary, Gopalnath sir, to give the vote of thanks.
So we have discussed a lot and spoken a lot. At the end, everything is so great, so wonderful. So which cannot be expressed in words. So I'm thankful to Professor Chipper and Dr. Urmi. Actually, she was so active all the time, and she's interacting and has organized so many lectures by herself. And she's one of the most active members. And Dr. Anand from the Hisar, she's also has done a lot of work. But the idea came in the mind of Dr. Vinod that it should be done online. And I was really so satisfied that something is being done by Dr. Vinod. And I knew that it is Dr. Vinod who can do it. So it was the great job. Actually, Dr. Vinod, during this COVID time, we have almost done 18 lakh sample testing in our lab. So I was so busy that I could not think that what can be done for the society. But in the meantime, you came like a boon, and things has been materialized. And all these 3 days was so good, so nice. And all these awards and these prizes which you have given is really flawless, and it is really encouraging all the members. My congratulations to all the achievers. And again, I would request those people who have presented and has not received the award this time, in future also keep on working, and I can assure you that if your quality of work is good, you'll be getting one day, definitely, because we are working, we are thriving, and we are the new society. So we'll be doing this one. And I'm thankful to my president, Dr. Chipper, whose guidance was already there all the time. And we used to discuss on telephonically so many times. Dr. Binod also, every time, constantly, we are rather shaping the certificates and invitation, all those things were there. And then Dr. Ramesh, so active person, and all those persons who chaired these different sessions and who worked as a moderators. I'm grateful to them also. Dr. Sangeeta, I congratulate you once again. You really presented so well the basics of bacteriophage work. So I never went to that extent which you presented. So I think that is essentially needed for us, because we are looking only the application part. So that is also very important. And again, I'm thankful to all the delegates who delivered, who participated, and who spoke, who delivered the guest lectures, invited lectures from outside the country and inside the country. And at the end, not speaking too much, only one thing, that we must give a big clap to Dr. Binod Kumar and his team for organizing such a wonderful conference. Thank you very much.
Thank you. Thank you very much, sir. Thank you. Thank you for all the support. Thank you for all the support. At the end, all the delegates will get a participation certificate by tomorrow. And the presenters also, presenters and award winners, you all will get a certificate by tomorrow evening. Thank you very much, sir. Thank you very much. And I request the life members to join for a general body meeting, which will be held in another 5 minutes. Please log in with the same, the link. Right. Thank you very much. Thank you very much. Thank you, Mark. Thank you for your support. Thank you. Thank you very much. Thank you.